Hi, my dear students. Today's topic is amino acids in health and disease. So, specific learning objectives at the end of the class, you must be able to define amino acid, to classify amino acids, define isoelectric pH, mention its significance, peptide bonds with characteristic as well as biologically important peptides. So first we should know what is meant by amino acid. Each and every topic I'm just insisting you to learn the definition and then go for other importance as well as functions. So amino acids are the monomeric unit or building blocks of proteins joined by specific type of covalent linkage. Here it is a primary protein structure which is a sequence of a chain of alpha amino acids. So single chain of amino acids composed together to form a protein. So next, chemistry of amino acids. First, you should know what is the molecular formula of amino acid. RCHNH2COOH. In that, two groups are there. One is carboxylic group, another one is amino group. So carboxylic group is nothing but COO minus and amino group NH2 plus. So both positive and negative compose together to form a protein molecule. Optical activity, one of the property of every carbohydrates, proteins, lipids we'll be telling. So in that L form and D form is there. Just nothing but D form, NH2 remains in the right hand side, whereas L form, NH2 stays in the left hand side. So left as well as right in the D form and L form shows it is a optical isomerism. In optical properties, except glycine, mostly glycine has a chiral group which has four different groups. What is meant by chiral group? In MCQ, they may ask what is chiral group. So four different groups are in between. It carries OH, H plus, other phosphate group or any other group, methyl, ethyl or any group which carries, that is called chiral or optically active carbon atom. Except glycine, every amino acid has asymmetric carbon atom. So normally glycine is a basic simple amino acid. So in nature there are 200 different amino acids but 20 constitutes of mammalian proteins. 20 are the only amino acids coded by DNA. In that 21st amino acid is selenocysteine. In MCQ they will be asking, and 22nd is pyrrolysine, which is a stop codon UAG, uracil, adenine and guanine, which is called a triplet codon. Importance of amino acids. It forms protein, biologically important peptide. This is very important question. Often they'll be asking in amino acid chemistry part. There's a name some biologically important peptides. Transport and storage form of ammonia. Example, glutamine will be involved. Buffering activity. As if in already a class, hemoglobin class I have told histidine, it acts as a buffer capacity. So next, detoxification reaction. Example, glycine, cysteine and methionine. Neurotransmitters, glutamate and glycine. So just know the name of amino acids and what are the roles played by it. You just know for importance of amino acids. And hormones, thyroid hormones, which is nothing but T3, T4. Precursors for other metabolites, glucose in gluconeogenesis, heme, etc. and glycine in bile salts, which is acting as a conjugating agent. So very important role played by amino acids in overall functions. Classifications, in this we have given many things: chemical nature, structure, nature or polarity, nutritional requirement and metabolic product of amino acid. In this, very important thing is nutritional requirement of amino acid, which comes under essential and non-essential amino acid. So that plays an important role for you in exam point of view. But other things I'll just tell a gist of it. Neutral amino acids, glycine, alanine, valine, leucine, isoleucine, serine, threonine, cysteine, methionine, proline, phenylalanine, tyrosine, tryptophan, aspartic acid and glutamic acid. So these are all comes under neutral amino acids, whereas acidic amino acids, aspartic acids and glutamic acids. Basic amino acids, lysine, arginine and histidine. Side chains also I have given. You just note it down. If they are asking, you can write it down. Aliphatic, glycine, alanine, valine, leucine, isoleucine. Aromatic, which is nothing but contain benzene ring, phenylalanine, tyrosine, and tryptophan. Mostly in exam point of view, they'll be asking aromatic amino acid inborn errors, disorders. They'll be asking in that phenylalanine important exam mark 5 mark question. Phenylketonuria they'll be asking. So hydroxyl group, which is nothing but OH, serine and threonine. Acidic, as usual, I already told. Basic, amino group, proline, MCQ they'll be asking. And sulfur containing amino acid also one of the important detailed question. Cysteine and methionine they'll be asking inborn errors of sulfur amino acids. Classifications overall hydrophilic according to polar and non-polar nature. Polar is charged particles, non-polar is it is not having any charged, whereas here negatively charged aspartic glutamic acid. Positively charged lysine, arginine, histidine. Uncharged serine, threonine, asparagine, glutamine, cysteine. So just you know the names of the amino acids for hydrophilic and hydrophobic I have given here. Aliphatic side chain as well as aromatic side chain. So note down the amino acids for polar and non-polar group. So these are all examples for just a view or information. And this is very important 5 mark question or sub questions they may ask in 2 marks also. What is essential amino acid? Name some essential amino acids. So when they are called as essential amino acids, they are not formed inside the body. And so it is essential to be taken in the diet. Example, mat will fly class they have told it seems methionine, arginine, threonine, tryptophan, valine, isoleucine, leucine, phenylalanine and lysine. Semi-essential amino acids, histidine and arginine, partially synthesized inside the body, but it is not sufficient for the functions. So histidine and arginine plays a role with semi-essential. In University Viva questions, they will ask name some semi-essential amino acids. Non-essential, they are not synthesized inside the body. Rest of the amino acids that are formed in the body in amount enough for adults and children. So no need to take through diet. It is synthesized inside the body. Glycine, serine, tyrosine, cysteine, alanine, aspartate, glutamate, proline and hydroxyproline. Other than essential amino acids, you can call as non-essential amino acids. Metabolic classification, this is very important 2 mark question. Glucogenic. Name some glucogenic amino acids. Name some ketogenic amino acids. Or both acting as a gluco and keto, they will ask. So note the amino acid name and write in the test. You'll get full mark for that. The derived amino acids, cysteine, hydroxyproline and hydroxylysine. You have read about the derivatives of monosaccharides when we have taken carbohydrate chemistry. So like that here also some derivatives of amino acids which plays an important role. Here desmosine and isodesmosine found in connective tissue protein as well as elastin which is a specialized protein. Gamma carboxyglutamate, it is a carboxylation of glutamic side chain occurs in many of clotting proteins. Example prothrombin. Phosphoserine, phosphothreonine and phosphotyrosine, they involved in many regulator proteins. Properties not much important but very important thing. One term you have to know is Zwitter ion. Before that I'll just tell a gist of it. Physical properties, they are colorless crystalline. Solubility, soluble in water, insoluble in organic solvents. Melting point I have given. Taste with a sweet as well as bitter taste I have given. What are the amino acids you have read at the +2 level I think. So chemical properties, due to ionizing property of amino acids, it exerts as acid-base behavior, ampholytes and buffering activity. So in this COOH carboxyl group behave as an acid that mainly donates, whereas NH2 accepts. You should know which is donors, which are acceptors. So COO minus are donors and NH2 are acceptors. Amphoteric properties, why they are called ampholytes? You will be knowing the term amphoteric, both positive and negative. Here it contains both acidic and negative groups. It can donate or accept proton. That's why they are called as ampholytes. Isoelectric, this is very important 2-mark question, or sometimes they may ask for 4 mark also in subdivisions. Better read that and write it neatly. This slide is more than enough. Acidic pH, amino acid is positively charged. Alkaline pH, it is negatively charged. So it carries no net charge and is known as isoelectric pH or isoelectric point. At isoelectric pH, the amino acid exists as a Zwitter ion and the solubility and buffering capacity will be minimum in that point. So in this particular thing, you have to draw sure for to get full mark. Both amino acids I have given here and Zwitter ion, COO minus and NH3 plus. How far it is obtained as a Zwitter ion, I have given this diagram. So this is very important thing. Substance having two-way property called amphoteric or ampholites. It contains both positive and negative groups. Some key terms we will be expecting in each and every answer. So fulfill that and we'll give marks for you. And this is titration curve of glycine. Sometimes they may ask what is the PI value of glycine, which is nothing but 5.97. Next, buffering action. In hemoglobin chemistry, I have told histidine serves as a best buffer at physiological pH due to its pKa value nearer to physiological pH 6. All the amino acids pKa value far away from 7, pH 7. So that's why mostly histidine serves as a buffering capacity for hemoglobin in the blood pH. And next, reactions. In this reaction, one important thing I want to tell you, transamination and deamination is very important. But before that, decarboxylation, very important role they are playing. Removal of carbon dioxide from histidine, it gives histamine, tyrosine to tyramine. So here tryptophan converted to tryptamine, lysine convert to cadaverine, glutamic acid converted to GABA. So why this is happening? With the chemical reactions, what we are going to do as an MBBS student, you should not think like that, because tryptophan converted to tryptamine, which involves as a neuromodulators, GI health, as well as neuropsychiatric conditions, schizophrenia patients, it will be elevated. So it acts as a marker also. And lysine converted to cadaverine, which involves as a microbiota, anti-inflammatory as well as anti-cancerous agents, and glutamic acid converted to GABA, which is a neurotransmitter in central nervous system. So amino acid derivatives, when it is converted by decarboxylation or dehydration or transamination, some reactions to take place in order to play a role in clinically significance. Here again, amide formation, aspartic acid combined with NH3 to form asparagine. You will be knowing this is amino acid, which comes under which group, you will guess it. Glutamic acid and NH3, glutamine. And next comes very important 5-mark question. From this, you have to note it down. Transamination, 5-mark question. Again, I'm telling it is very important. You should write the definition first. Alpha amino group of amino acid is transferred to a keto acid to form new amino acid and its corresponding a keto acid. So this is called transamination definition. You should write, no need to write the structural formula, but write the word alpha Kg plus alpha L-amino acid gives out L-glutamate plus alpha keto acid. So one amino group and one amino group as well as one carboxyl group combine together to form a new keto acid with amino acid. So this is called transamination enzyme, transaminase with pyridoxal phosphate as a cofactor, it is involved. So if there is any deficiency, pyridoxal phosphate, you can guess which vitamin. So if there is deficiency of PLP, transaminase won't work. If transaminase is not working, this reaction doesn't happen. If this reaction is not taking place, some problems, some disorders will lead. So here urea formation happens in the liver. Transamination acid is irreversible. They also help in synthesis of non-essential amino acid. So very important role played by amino acids in transamination reaction. Next, oxidative deamination, you can combine this also. Glutamate dehydrogenase, amino acids removed to form keto acid and ammonia. So glutamate converted to alpha Kg plus NH4 plus. So here in the body, glutamic acid is the one to undergo oxidative deamination. Next, carbamino compounds I have told in hemoglobin chemistry class, carbamino hemoglobin, which forms carboxy amino hemoglobin that gives carbonic acid, and that carbonic acid will be giving H plus HCO3. I have told as a Bohr effect and chloride shift in the previous class. So better learn that, just interact with them and then read as well as write it. So next, transmethylation, other reactions. These are all but not much important, but you should know methionine with acceptor, it forms methylated acceptor plus homocysteine. This is very important marker for cardiac functions. Cardiac failure, it will be elevated. So in order to find as a marker, we can use this. Homocysteine comes from methionine by transmethylation reaction. So these are all very important reactions that takes place in our body. Reactions of amide groups, glycosidic bonds will be formed. Glycosidic bonds is nothing but cardiac glycosidic bonds which plays an important role as antibiotics. And reactions of SH group, here sulfur hydryl bond plays an interactive role with cysteine and cysteine. So next, characteristics of peptide bond. Here just I want to tell the characteristic features. COOH and NH group. You should know this is a peptide bond. And very important, another 5-mark questions, biologically important peptides, hormones, they'll act as a hormones, antioxidants, antibiotics, anti-tumor drugs, sweetening agents. So I've given the example as well as in shorts also I've made in my YouTube channel. Go and read, write neatly in the exam. These are all important, biologically important peptides I've given: aspartame, carnosine, thyrotrophin releasing hormone TRH, glutathione, very important antioxidant playing in our body, acting as a tripeptide with 3 amino acids. These you have to note it down: glutamic acid, cysteine and glycine. What are the 3 amino acids composed to form glutathione? They will ask, note it down. And biologically important, it suppresses oxidative stress. So as an antioxidant activity, how far it plays? Glutathione peroxidase, integrity of RBC membrane. These are the key roles, key words. You have to note it down and write in the answer. These are all additional information: angiotensin, pentapeptide, methionine, enkephalin. If you want, you just read it. Angiotensin you will be knowing for blood pressure, as well as they'll be used as an analgesic. And then bradykinin with oxytocin I have given as a clinical function, posterior pituitary, and it is a powerful vasodilator. Functions alone, you just note it down. These are all very important, biologically important peptides. Vasopressin, very important antidiuretic hormone. With gramicidin I have given, act as an antibiotic. So thank you, my dear ones. You have asked amino acid chemistry class and I have given. If there is any queries in this class, you can ask me through in the channel itself. Thank you, dear.